like to welcome to the stage Alex Shephard, Market Manager for Cell Therapy at Unchained Labs. Thank you.
Hello everyone, hope you're all having a good meeting this week. Personally, it's been really interesting over the last day or so to hear about what you guys are all doing and really see the progress that EVs are making towards the clinic, really. Now, I've been in the EV exosome field for around a decade now, and it always seems as though they're labeled as the next hot thing and talked about really enthusiastically, but up until now, they haven't had that breakthrough moment, potentially, for therapeutics that some of the other delivery mechanisms have had. So hopefully, based on some of the work being done in this room, that can change and they can move forward in the field. But however you're using EVs, whether that's from a kind of core research perspective or for diagnostics and biomarker analysis or as an engineered delivery mechanism, we all know that despite their great potential, they can be a real challenge to work with due to several different aspects of them. So obviously, very often you're working with a complex sample, whether that's a biofluid for diagnostics or whether it's just a crude cell culture sample as part of your production process. There are a lot of other particles in there besides your EVs. So protein aggregates, cell debris, liposomes, lipoproteins, etc. So if you're trying to analyze your EVs and actually quantify them and interrogate them, it's very hard to be sure that you're actually looking at the EVs rather than other aspects of your sample. Secondly, obviously, they're really small. We've talked about sizing a lot already today, and I think it's becoming more abundant and more clear that not every EV is 100 nanometers. There's a large distribution of smaller particles based on some of the recent EM data, for example. And if we're just looking at kind of 80, 90, 100 nanometer plus, probably missing a lot of important particles in your analysis. Also highly complex. No two EVs are the same. Very heterogeneous samples, which means if you're trying to analyze them using some of the classical techniques for other vectors, such as DLS, for instance, you're not going to be getting a real analysis of your sample. And then finally, again, in a lot of instances, particularly if you're on the diagnostic side of things, you're often looking for a really rare population, perhaps a couple of EVs per 1,000 or even per 10,000 or more of your population. So all of this means that you really need a single particle technique that is both really sensitive in terms of sizing limitations and in terms of concentration to be able to accurately figure out what is in your EV sample. And even amongst the abundance of single particle techniques that there now are for EV analysis, a lot of them can struggle with the unique complexity of these particles. So for instance, you may be doing a more general analysis that isn't specific to EVs via NTA or similar because of the limitations that there can be with that fluorescent analysis. And generally, most techniques, their sizing capabilities start to cut off around 50 or 60 nanometers, which, as I just mentioned, you're missing a lot of those smaller particles. And what is generally common amongst all these techniques is that the quality of the data that you get is only as good as the purity of the sample that you're able to put into it. So again, a particular issue for anyone working with biofluids of any sort, because you've normally got a small starting volume, and you've got to then use some of that to purify and isolate your EVs. So also that isolation techniques are normally in some way going to skew your analysis due to whatever the biases of that method are. And even if you're working in cell manufacture and EV production, it makes it hard to look at the more crude upstream samples. So that can make in-process monitoring a challenge because it's hard to know what you're really starting with, so you can't perhaps properly judge the effectiveness of some of your different purification steps. So I'd like to introduce you to Leprechaun, which is Unchained Labs' exosome and EV analysis platform. Leprechaun is a single particle analysis technique which doesn't require purification beforehand. And it can measure concentration, size, and the phenotype, both externally and internally, of your EVs. So some of you may think that this sounds a little bit familiar, and that's because Leprechaun is the third generation of the ExoView platform, which was made by NanoView Biosciences. So in 2022, Unchained Labs acquired NanoView, and Leprechaun is now the third-gen, up-spec'd, redesigned version of the Exoview technology. So we have Exoview has a large academic and industry-focused customer base, and Leprechaun is now the new version of that. So how does it work? Well, it has a consumable chip called the Luni. This is pre-coated with highly specific antibodies, either against the tetraspanins, or as we'll see in a minute, you can customize that for specific markers of interest. And once you immunocapture your sample, the instrument then sizes up each individual particle using single particle interferometry. So it's a little bit different to some of the other optical techniques. This is a little bit different to NTA or scattering as on flow cytometry. But it means it's really sensitive both in terms of the limit of detection and in terms of the sizing resolution. And then we combine this sizing with a free color fluorescent microscope. So we have red, green, and blue channels where you can stain your EVs for different markers or look at intrinsic fluorescent tags that you may have engineered onto your EVs. So a little bit more about the chip itself. As our standard human chip comes with CD9, CD81, and CD63 pre-coated. And then every chip has an inbuilt negative control. So you can see if there's any background, any non-specific binding, that is accounted for. And each of these antibodies is spotted in triplicate. So you've got your technical replicates in one chip. You don't have to run multiple chips. And we have other chips that are already pre-coated for plasma. So for example, if you're interested in platelet-derived EVs, there's a CD41A spot that you can use. Or if you're working with murine samples, we have an equivalent tetraspanin chip for your mouse samples. In terms of the sample prep, it's really hands-off. So it's a little bit like an ELISA process in terms of capturing, washing, and then staining your samples before you actually use the instrument itself. But all of this is automated using our Luni washer. So this allows you to wash, stain, and dry up to 24 samples in one go. So as I say, hands-off, so it's saving you time. Also, you don't have to worry about user-to-user variability. And then when it's acquired all this information, it basically uses colocalization to build up a picture of your EVs. So we'll overlay the 3 different fluorescent channels and identify particles that are single, double, or triple positive for whatever markers you're staining for. And then in terms of the sizing, as I say, each particle is sized individually to build up that size profile. And the big difference of Leprechaun compared to its predecessor, the Exoview, is that we've lowered that lower sizing limit. So Exoview could size down to 50 nanometers, as I say, pretty comparable with some of the other technologies out there. Leprechaun can now go down to 35. And I think you could see that by looking at Exoview data previously, you'd often have this peak that you're kind of seeing cut off at that 50 nanometer point. And now with Leprechaun, we can actually see the majority of that peak. And what we found is that the sizing aligns really well with what you tend to see from EM. So there seems to be a lot of small stuff as well as that larger 80, 90, 100 nanometer particles. So we're now able to kind of open the door and get quantification of the whole range of EVs from exosomes down to exomeres. So by combining the sizing information with the fluorescent imaging, I say we can build up a picture of your sample, and you will get readouts on concentration, the phenotype via colocalization analysis, and a size profile. And this is all on a marker-by-marker basis. So you'll get a different size distribution for, say, each of your tetraspanin populations, or for all of your custom markers that you may have looked at. And all of this data is from one run of the Luni. So it's one scan for all of these different outputs. In terms of sample requirements, as I say, it is a really sensitive technology. You need a maximum of 25 microliters, and that's whatever kind of sample you're using, however pure it is. But in most cases, most samples, you're probably looking at less than 5 microliters very often. So low sample requirements, no need to prior purify, and as I say, compatible with cell culture. I think we've tested almost every biofluid there is out there. So it's always interesting to see what people are working on. But yeah, all biofluids seem to be compatible. So in terms of the data and the applications that you can use this for, I think the thing most of our customers are interested in is looking at specific subpopulations of EVs.
And we can differentiate and provide concentrations and sizing for each of the different subpopulations present. So this is an example of the sigma GFP labeled EVs, whereby we can separate out the GFP positive from the GFP negative and see that we actually see some size differences, whereby the GFP negatives are much smaller, so whether that's due to their biogenesis or whether it's the labeling of the GFP itself that's making that difference, we can start to really break down and analyze the subtle differences that there are within the sample. And as I say, it's not just limited to external markers. We can also look inside your EV at the cargo. So in this instance, we've probed for syntenin, which, as we've just heard, is one of the general markers of EVs. And we can actually get inside with a light fixation and permeabilization and pick up your protein within that. Or, as I say, you can customize that, look for any cargo that you want to look for, or if you've got a tagged GFP or RNA inside, then you can also pick that up. And a nice example of this is from a recent paper from one of our users, where they were loading their EVs with doxorubicin as the cargo, and looking at the intrinsic fluorescence of this relative to a tagged CD63, they were able to show co-localization of their doxorubicin cargo with their EVs, and actually measure the loading efficiency of their EV system. And we're not just limited to protein cargoes. So there's been some interesting recent work from Johns Hopkins University, where they've been looking at sequence-specific RNA detection using the ExoView versions of this platform. And using a small molecule fish probe, they were able to detect specific RNA sequences that they had loaded into their EVs. So, as I say, there's a lot of interest in miRNAs and EVs as a delivery mechanism for both RNA and DNA. So this can also be used to check that you're getting your nucleic acid inside your EV, as well as any protein cargo. So as I say, I've talked about the tetraspanning kind of analysis and capture, but you can customize this if you have a specific protein or very often a biomarker that you want to specifically look at that EV population. So we have a set of Flex consumables, which are basically chips where you can add an antibody against any marker that you want to in your own lab, and customize the capture step to look at that population of EVs. And by doing this, one of the interesting things we've seen is that it can also be a good way to check if your protein or if your marker is EV associated or soluble. Because obviously, that's one of the advantages of single particle analysis techniques, is it can be used to verify that this protein of interest is actually on an exosome. It's not just free in solution. So in this instance, we customize the capture step to be against PDL1. And what we see on the left-hand side with our hex is they're a negative cell line for PDL1, but the HMEX do express PDL1. And so then we fluorescently stained with antibodies against PDL1 as well. So we're capturing and staining with PDL1 versus capturing and staining against the tetraspanins in the blue. And so by using that second step, we can say that, okay, we see a lot of PDL1 signal in the green bar, but actually the amount of that is associated with tetraspanins, which is in the blue bar, is really low. So in this instance, although there's PDL1 released from the HMEX sample cell lines, it's really not EV associated. It's soluble protein instead. So it can be a really useful check to establish where your protein of interest is. As I say, it's an incredibly sensitive technique, so we can detect down to 5 times 10 to the 5 particles per mil in terms of EV concentration. Again, that's from crude and pure. There's no impact on the sensitivity regarding the sample type, and it can handle, as I say, any sample type. So in terms of the reagents, we sell kits of these chips. You get 8 chips in a kit, so that's 8 samples. It's 1 sample per chip. You get all the buffers required for the washing steps to use with the washer that we mentioned earlier, and all the cargo reagents if you want to look at internal proteins. And we also provide you with the tetraspanin fluorescent antibodies as standard, and the syntenin antibody as well. But as I say, any antibody you have in your lab, you can customize that with as well. Software is incredibly easy and straightforward. So I think we have a great software design team at Unchained, and they've made it really easy to use. So there is automated gating in terms of the fluorescence and the background. So anyone familiar with a lot of, say, flow-based systems, you have that manual gating step, which not only is a pain, but can also introduce a lot of variation in terms of what is my positive and negative signal, where do I set that threshold? Well, the software will do it for you automatically. So if you want to tweak that and play around with it manually, you can do, but all of that stuff is standardized. Again, really helping to reduce user variation and also save you time. So it's literally load up, get your results, very minimal analysis time. So we have, as I say, a variety of customers and a large user base, both in the academic world and also in pharma and industry. And they're doing some really exciting stuff with this platform. So, for example, Direct Biologics are using it as part of their QC process in their current phase 3 clinical trials for their exosome therapeutics. And we have a range of applications from R&D through to process development and also analytical development. So yeah, I just wanted to introduce you to Leprechaun. As I say, if the all-round platform for figuring out exactly what's going on with your EVs, regardless of whether you're in that core research, biomarkers, or EV engineering. And happy to take any questions. We also have a booth out in the foyer, which I'm sure you've seen. It's incredibly bright, but as are the shirts. But thank you for your time, and I hope you enjoy the rest of the meeting.
Does anybody have any questions for Alex? Christopher.
Why the name Leprechaun?
That's it, because we have a great marketing department. I mean, if you want to find a link, everyone asks if it's an Irish company. It's not, but the technology was originally developed in Boston. So if you're being tenuous, there is a bit of a Boston Irish link in the Celtics. So that would be my explanation if you want a reasoned argument. But all of our products have to have UN in the name, so there's also that tie-in.
I had a question regarding the using the 3 different colours and the laser. Is there any issue with cross-signal contamination?
Lead through the signal, that kind of thing. Yeah, so I mean, it's always going to depend slightly on what fluorophore you're using. So the ones we use in the kits, we use the CF dyes, which have like the Alexa dyes, have a very narrow excitation and emission spectrum. So as with any fluorescent-based technique, you have to be aware of what dyes you're pairing together. Something like PE, for instance, have a much broader spectrum. You may get a limited level of bleed-through, but each channel is excited individually, from lowest energy up to highest energy, which should minimize that. But yeah.
Is it a filter bait, or is it like a okay?
Yeah. So we have a filter set for all of the different channels. So that.
Does anybody else have any more questions? In which case, we'll thank you very much, Alex, and that concludes this morning's session. So we just give a round of applause for Alex.